Hello, this is the third of a series of videos I'm doing on the topic of DNA. DNA at upper sixth is a very big topic. Often schools will just do all of it as in one booklet, suggesting that it's just one big topic. But I find that it's best to split it into three parts, which are fairly easily distinguishable. And so I've already done videos on the first two parts. The first part of this topic, the DNA topic, would be about the nature of the DNA code and protein synthesis, DNA and protein synthesis, and I would include in that topic then also some discussion about mutations and also epigenetics. So that's the first topic, DNA and protein synthesis. The second part of it, in the second part, I put everything under the heading of DNA technology. And in that section, in that video, I looked at the various techniques involved in working with DNA, manipulating DNA, cutting DNA, amplifying DNA, and so on. So that topic would include restriction enzymes, DNA probes, microarrays, polymerase chain reaction, DNA fingerprinting, markers, etc. So those things are all together in that second topic, DNA technology. And then the third topic, which sits, third part of it, which sits largely on its own, I would include these two items here, gene transfer and gene therapy. And even on its own, this is quite a big and long topic itself. So this is the topic I want to try and look at today in this video, but I suspect that I'll only get the gene transfer bit completed today in this video, which should last about 45 minutes or so. Gene therapy, it may take a further video to look at that. So as with all of these videos, you'll really need a set of my notes in front of you. Since I constantly refer to the notes and refer to the content as I go along. You also need a set of past paper questions, which I refer to as I go through the content, and make reference also then to the appropriate past paper question, and as far as possible try to do that question in the light of the content. You'll also then have a set of answers yourself, and you can go through these questions then alongside the video. If you need any of these resources, then my mobile number, is on the screen there, and you can get in contact with me. I'll get you some materials. Okay, so this topic we're doing today, gene transfer, which is essentially about the transfer of genes from one organism to another organism, or maybe just into an organism. How do you transfer genes into we're going to look at how you transfer genes into bacterial cells, into plant cells, into animal cells. So then we're going to finish off then maybe looking at gene therapy. So okay, this topic then, the first part of this topic essentially looks at the transfer of genes into a bacterial cell, and the exam questions that are appropriate here would be the past paper questions that I've indicated here would be question 1, 1B, 2D, question 4, and 9, the essay question, question 9, the essay question. And question 9 basically covers all of what I'm going to be doing today. That's an essay question on this topic. So I'm just going to then start into it and discuss some questions as we go along. So the gene, when we're talking about transferring a gene from one organism to another, or transferring a gene into an organism, we often refer to that gene as the donor gene, and we refer to the cell or the organism into which the gene is going as the host cell or the host organism. So we want to transfer a gene into another organism. Let's say we want to transfer a human gene into a bacterial cell. And that's a situation you came across at GCSE. You might remember that you could transfer a gene for insulin production, a human insulin gene, into a bacterial cell. And once you transferred the human gene into the bacterial cell, the bacterial cell acted on the gene. The gene was expressed inside the bacterial cell, and you got the protein for which the gene coded. And at GCSE, the gene that you looked at was the gene for insulin. So you're able to put an insulin gene into a bacterial cell, and then you grew the bacteria up. You multiplied the bacteria up so you had millions of them. And then all of those bacteria then produced the protein insulin, the hormone insulin, and you could extract the insulin then and use it to treat diabetes. And that nowadays is the source of all of our insulin from this process. So essentially, what I'm going to look at today is just look at the whole business of obtaining a human gene and transferring it into a bacteria. And that will probably occupy the whole of this video. So I've broken it down there on the first page of Search. You can break this down into stages. And the stages are, number one, you have to obtain your human gene. So get your human gene first of all. So where do we get our human gene? Then when you've got your human gene, you have to insert your gene into what's called a vector. Well, often you'll get asked the question, what is a vector? A vector is a mechanism or means of transferring a foreign gene into a host organism. So it's a mechanism or means transferring a foreign gene into a host organism. With regards to bacteria, the best way to get a gene into a bacterial cell is to take out the bacteria's plasmid. Take out a plasmid out of the bacteria. Essentially, the plasmid represents the bacteria's chromosome. It's not a chromosome, but it's like a kind of a circular chromosome. So it's a circular section of DNA. And you take that out of the bacterial cell, then you can insert the human gene into that, and then you can put the plasmid back into the bacterial cell. So in essence, the plasmid has acted as the gene carrier, or the plasmid is acting as a vector. So inserting the gene, if you insert the human gene into the plasmid or into the vector, then you have to get the vector, you have to get the plasmid back inside the bacteria. So you put the plasmid back inside the bacteria, and then lastly, you have to identify those bacteria that actually contain the plasmid with the human gene present. And this is possibly the most difficult part of this section, because in the whole process is not very successful. In essence, what we're saying in essence, what we're saying is, if you take a gene, so you basically take your bacteria and you take out its plasmid. So there's its plasmid there. In case that's the plasmid. Take the plasmid out of the bacteria, and you remember this from GCSE. Take out the plasmid, there's the plasmid there. You then cut open the plasmid, put the human gene into it. Put the human gene into the plasmid, and then you put the plasmid back inside the bacteria. So we'll just put the plasmid back in with the human gene in it there. Okay, so that was the process at GCSE. Take the plasmid out of the bacteria, put the human gene into the plasmid, put the plasmid back inside the bacteria. All sounds very easy. It's pretty straightforward, but in essence, the process is not always very successful. So I'll just rub this off. It's not always a totally successful process, even though the notes and the text and that suggests that it is a very successful process. So those are the 4 stages, and essentially the essay question, I think it's question 9 and question 1 follows through those steps one after the other. So question 1, I'm going to leave out some parts of it, and I'm going to leave out the easier parts, and I'm going to focus on the more difficult parts. So the first thing, the first part 1B asks is 1B1. Describe 2 methods by which a specific gene could have been obtained from human cells. So 1B1 is asking us, how do you get a human gene in the first place? And really there's 2 methods. And the 2 methods, I'm going to describe them very quickly because generally they just deserve 2 marks each. So you see there's a total of 4 marks there. So you get 2 marks for each of the 2 methods. And the first method involves cutting the human gene out of the human DNA using a restriction enzyme. You would use a restriction enzyme that cuts with sticky ends, and I looked at sticky ends and such an enzyme in the previous topics. You need to look back at that. So you cut out the human gene using a restriction endonuclease enzyme. It cuts with sticky ends, and then you can locate that gene. Once you've cut the gene out, a gene will be in a section of DNA. You can locate that gene using a DNA probe. So that's the 2 marks for that. 2 marks for one method, for a method of getting a human gene. Cut the gene out with a restriction endonuclease enzyme, and then locate the gene, locate the piece of DNA that contains the gene using a DNA probe. That's the first method. The second method is to use messenger RNA and reverse transcriptase. And this takes quite a bit of explaining. It really only deserves, you'll see there, it only gets 2 marks. So, but it can take ages to actually explain it. So this is another way of getting a human gene. Let's say you want the human insulin gene. Well, if you want a human insulin gene, you could go to a human cell that's producing insulin. That's a pancreas cell. So there's a pancreas cell. And inside the nucleus of the pancreas cell, there's the nucleus. If you can see the nucleus there, inside the nucleus, you've got the DNA, which is, so here's the DNA inside the nucleus, let's say, if you can see that. And what we know is that one section of that DNA contains a gene, the gene for insulin. And then what we know is that, say the gene for insulin's there, so that's the gene. You know then that the gene is copied onto what's called messenger RNA. And then the messenger RNA leaves the nucleus and goes out through the nuclear pores. So the messenger RNA, I've just put it in red there, it can leave the nucleus and then it goes out to the RER. Yeah. So the RNA, which is essentially a copy of the gene for insulin, leaves the nucleus and goes out into the cytoplasm. And that's the basis of this second method. If you go into the cytoplasm of this insulin producing cell, this pancreas cell, and you find the messenger RNA out here, you can take the messenger RNA and you can work backwards from the messenger RNA. So first of all, you use an enzyme called reverse transcriptase, and it will work you back from the messenger RNA to the coding strand, or the coding section of the gene, the coding strand section. And that's the first mark. So the first mark is to use an enzyme, obtain the messenger RNA from the cytoplasm of the pancreas cell, and then use an enzyme called reverse transcriptase, which will make you the coding strand, or the single strand of DNA from which the messenger RNA was made. Then you use a second enzyme called DNA polymerase, and that will make you the other strand, the non-coding strand. And when you have the 2 strands of the gene, then essentially you have the gene at that stage. So that's the second method. So to summarize that for 2 marks, what you say is use messenger RNA from a cell plus an enzyme called reverse transcriptase. That'll make you single-stranded DNA, and then you use the single-stranded DNA along with another enzyme called DNA polymerase and free nucleotides, and you can make the double-stranded DNA, and that effectively is your gene. So that's the 2 methods then. 2 methods to get the human gene, the human insulin gene in this case. So we can just take that off the screen. Yep. And if you're following the essay, which is question 13, you'll see essentially that's the essay is asking the same thing. Question 13, the essay asks you, we've got it right here. Question 13, it says, describe the processes of obtaining the desired genes and their subsequent transfer into the cells of organisms. Well, at this stage, we're just looking at transferring genes into bacterial cells. So we're just looking at bacterial cells so far. We'll come on to look at plant cells analysis. So those are the 2 methods, and that's dealt with on pages 2 and 3 in the notes there. Now, in the next stage is once you have the gene, you got the human gene, you want to insert it into a vector. And the vector, of course, is the plasmid. So essentially, there's the plasmid that you've taken out of the bacteria. And what you want to do, and this is the human gene. Let's get it maybe in red here. So basically, what do you do? You cut open the plasmid, and you insert the human gene into the plasmid, like so. and then you put the plasmid back inside the bacteria. Now, there's a little complication here, and it's useful at this stage to look at question 1 there. And you'll see on the very front, I'll see in question 1, you'll see down the bottom of the page there, you've got 2 plasmids. Now the plasmid that we take out of the bacteria for this process, and you'll see there, top plasmid, you'll see the plasmid may contain a gene for resistance to an antibiotic called streptomycin. And it may also contain a gene for resistance to the antibiotic tetracycline. So this is the kind of plasmid we use. In GCSE we just drew the plasmid like that, but now we're realizing that plasmids are actually contain genes, the genes that the bacteria has. And this plasmid contains a gene for resistance to the antibiotic streptomycin, and also a gene for resistance to the antibiotic tetracycline. And what I've done is I've drawn a bacteria around that top plasmid there, and I would normally ask the question, I would draw a bacteria, and I would ask the question, you see that bacteria there with plasmid number 1 in it, what would that bacteria be resistant to in terms of antibiotics? And you can see there that if the bacteria has those 2 genes, then it's resistant to both those antibiotics. So there's how a bacteria can be resistant to antibiotics. It simply contains genes for antibiotic resistance. So if the bacteria contains plasmid number 1, it's resistant to both antibiotics. Now you'll see the plasmid down below. You'll see what has happened there. The plasmid down below has had a human gene inserted into it. And you'll note that the human gene has been inserted into the middle of the gene for resistance to the antibiotic streptomycin. That means when you put the human gene into the middle of that other gene, that disables that other gene. So that means, and I've drawn a bacteria around the bottom plasmid. So if you were to put a human gene into the middle of the gene for resistance to streptomycin, there's our plasmid with the human gene in there. If you put that plasmid into a bacteria, then the bacteria is only resistant to tetracycline, which is the bottom gene here, which hasn't been disabled, hasn't been damaged. But it's not resistant to streptomycin because the human gene has been, the cut has been made in the middle of the streptomycin gene and the human gene inserted. Now you'll see why this is relevant later on. You'll see this is a mechanism, this is a way of finding out which, or locating those bacteria that actually contain the human gene. Because if a bacteria contains a human gene, the bacteria will contain that plasmid. The bacteria then will only be resistant to tetracycline. It won't be resistant to streptomycin. Okay, so this is the kind of plasmid then that's used in these circumstances. So inserting the gene, inserting the gene into the bacteria. So you cut open the plasmid, and you cut open, according to that diagram there for question 1, you cut open, say you cut open the plasmid and you cut open the gene, you cut it open at the gene for resistance to streptomycin, and you insert your human gene in there. Then you now refer to the plasmid that contains the inserted human gene. You'd refer to it as a modified plasmid, or another term is a recombinant plasmid, or a modified plasmid. Now, what's the stages in putting the gene into the plasmid? Well, you'll see there, question 1, 1b part 2. It says, describe how the human gene could be inserted into the plasmid to produce the recombinant plasmid. And basically what you say is, the gene is cut open, the plasmid is cut open using the same restriction endonuclease enzyme that was used to cut out the human gene. By the way, we're assuming here that the human gene that's going to be inserted into the plasmid has been obtained in the first place by cutting it from the DNA using a restriction enzyme. So your first point here is you say, cut open the plasmid with the same restriction endonuclease enzyme that you used to cut out the human gene in the first place. In that way, the human gene and the plasmid DNA will have matching sticky ends. They'll have matching sticky ends. And you can then insert the gene into the plasmid, and you can get the gene to attach to the plasmid DNA using another enzyme, an enzyme called DNA ligase. So that's the third mark. So the 3 marks there for that. The plasmid is cut open with the same restriction endonuclease enzyme. You have to say same. If you don't say same, you don't get it. which provides, you would say, complementary sticky ends, or sticky ends that match the gene, and then the gene is inserted into the plasmid using DNA ligase, and that's the 3 marks. And so at that stage, then you should have what's called a recombinant or modified plasmid. And according to our based on our content here on this question 1, we should then have the bacteria that contains the plasmid, the modified plasmid, and the bacteria has now got a gene that makes it resistant to tetracycline, but the gene for streptomycin has been disabled, and then it also contains the human gene. So once you get the gene into the plasmid, the next stage then is reinserting, getting the plasmid to go back inside the bacteria. And you'll see there on question 1 again, 1C part 1. It says, explain how the bacteria might be encouraged to take up the plasmid, take up the recombinant plasmid. And this is the way they ask it, how would the bacteria be encouraged to take up the plasmid? In other words, basically, how do you get the plasmids to go back inside the bacteria? And the answer is, it's like a 2-mark answer, but usually they just give 1 mark. And they say, you place the bacteria and the recombinant plasmids in an ice cold calcium ion solution. And then you do something called heat shock. Basically, you start them in ice cold calcium ion solution, and then you raise the temperature quickly to about 40 degrees centigrade. And apparently that modifies the cell wall of the bacteria. It makes it more permeable and allows the plasmid to go back inside the bacteria. It allows the plasmid to go back inside the bacteria. Now, that is difficult to appreciate. How would you get a plasmid to go back inside the bacteria? Well, you know, GCSE would have said, say that was the bacteria there, and that was the plasmid. You put the plasmid back inside the bacteria. But the reality is, when we discovered that the plasmid isn't this size relative to the bacteria, plasmids are actually, I'm going to make one, microscopically small, so plasmids are really tiny. We just draw them with big circles like that because we can see what we're looking at, but they're actually microscopically small. And so you can imagine then maybe a plasmid going in through the cell wall and cell membrane of the bacteria. So that's it for 1 mark. How do you get the plasmid into the bacteria? You place the plasmid on the bacteria, modified plasmid, or the modified plasmid, or recombinant plasmid, and the bacteria in an ice cold calcium ion solution, and then you raise the temperature. This is referred to as heat shock, and usually you get 1 mark. Now, the next part is the most difficult bit to explain. I always find it very, very difficult to explain it, but students always want to know how it works or how it happens. And you'll see it there. You'll see what the question gets asked. It's in the question booklet. It's question 1C part 2. Question 1C part 2. 1C part 2. It's also covered quite well in another question there. This is a much older question. This other question, I think it's question 4. And possibly you might use question 4. That might be a question that you'd use in your notes, your own school notes, possibly, where you might have looked at question 4 as the basis for describing all of this. And of course then the essay question, I think it's question 13, the essay question. And then not question 13. Let me get this right. The essay question is question 9, and it also deals with this as well. But firstly, what I want to say to you is, if you look at question 1C part 2, here's the relevant bit. It says, outline the procedure by which the bacteria that have taken up the recombinant plasmid might be identified. How do you identify those bacteria that have taken up the recombinant plasmid? And you see, up until this stage, you might have imagined that this was always very successful. You took out the plasmid, you put the human gene into the plasmid, you put the plasmid back inside the bacteria, and everything was successful. Everything worked. But if you're looking in my notes there as you go along, you'll see on a couple of occasions I've said that various processes have very low success rates. So the first process that has quite a low success rate is inserting a human gene into a plasmid. So you'll see there on page 5, about 3/4 of the way down, I've said, note the process of putting a human gene into a plasmid has a very low success rate. I've said some very few plasmids actually will accept the human gene into them. And so at the end of the process, you might start with, say, 1,000 plasmids and 1,000 human genes, and you might only end up with one plasmid which has accepted a human gene. The other plasmids remain just as they were. They're non-recombinant plasmids or non-modified plasmids. You might only end up with one plasmid that's modified. So that's an important point. That step is actually quite difficult to carry out, and most of the plasmids at the end of that step, most of the plasmids you're working with aren't recombinant plasmids. And then later on, if you turn over the page then to page 6, I've said the process of getting plasmids to go back inside bacteria also has a very low success rate. Only about one bacteria in every 10,000 actually will take up a recombinant plasmid. Most bacteria don't take up a plasmid at all. So when you're trying to get plasmids to go back into bacteria, it's not a very successful process. Most bacteria won't accept the plasmid. And the bacteria that do accept a plasmid may end up with a plasmid that hasn't been modified. So I've said there, most bacteria either take up no plasmid at all, they don't take up plasmid, or they might take up a non-recombinant plasmid. And they're more likely to take up a non-recombinant plasmid because there are far more of them present. So it's at this stage then we want to discuss this. So usually I do it this, I try to illustrate it this way, and it's not always very successful, but it may work. So first thing I would say is, imagine all of this process is happening in a big beaker. In other words, in the big beaker, you're going to have plasmids, bacterial plasmids, and the first thing you're going to do is you're going to try and get a human gene inserted into a bacterial plasmid. And note I've got note the plasmid then, and I think I've got a slightly different version of this in my notes, but we're using, I think we'll use question 1 as our illustration there. And so what I'm going to do is I'm going to start off with, you'll see in question 1 on the front page, I'm going to start off with 2 of these plasmids which are indicated here, number 1 plasmid. So these are the unmodified plasmids. And there they are there. I've got them in the beaker. And what have we said? Each of them contains a gene for tetracycline, which is the bottom here, and a gene at the top for streptomycin resistance. Sorry, I should have said tetracycline resistance and streptomycin. So let's start off with 2 bacteria like that. Now, the first thing we said was that we wanted to insert a human gene into the plasmids. Now you'll see why I just do 2 plasmids but here, let's say you add into this beaker, you add in the human gene. So here comes the human gene. And we want the human gene to insert, in fact, you add in lots of human genes, loads of human genes. You add in multiple copies of the human gene. And basically you want it to go into a plasmid. Let's say this happens. Let's say the human gene gets inserted into this plasmid. So I've inserted there, and you see where it's gone in. It's attached, it's gone into the plasmid, and it's inserted into the middle of the a gene for streptomycin resistance. So we would say that this is a modified plasmid, yeah, or a recombinant plasmid, and this other plasmid represents the plasmid that hasn't taken up the gene, hasn't taken up the human gene. So this would be a non-recombinant plasmid. So what the situation I have at the minute just represents what happens essentially after you try to insert a gene into a plasmid. You end up with some plasmid. You end up with a very few plasmids that are recombinant plasmids. The majority of the plasmids are like this. They're just the original plasmid with its 2 antibiotic resistance genes in it. It hasn't been modified at all. Now, the next thing we want to do is want to introduce the bacteria. And this is, by the way, this is just an illustration. It's just my way of trying to explain this. And we're going to look at a situation. We're going to say, here's the bacteria. And we're going to imagine, I'm going to put 3 bacteria here. So 1, 2, and 3 bacteria. And we add them into the beaker. So we're putting these 3 bacteria into the beaker. And we're going to call them bacteria A, B, and C. Okay. So these represent just bacteria. You'll see why I've chosen to add 3 bacteria into the beaker. The idea now is that we want that plasmid, that kind of plasmid, a modified plasmid, to go into a bacteria. But what actually happens is you get at the end of the process, after you've added in your bacteria, notice your bacteria have no plasmids in them. At this stage, they're basically going to take up a plasmid. 3 things, you can end up with 3 types of bacteria. Let's go with bacteria A. Let's say bacteria A, remember all this process is very low, has a very low success rate. Let's say bacteria A, it refuses to accept a plasmid at all. So it doesn't take up either this kind of plasmid or this kind of plasmid. Now, if I was to ask you then, what would that bacteria be resistant to then? Well, it hasn't taken up either the original plasmid, that would have given it resistance to both antibiotics, or it hasn't taken up the modified plasmid, which would have given it resistance to tetracycline, but not streptomycin. It doesn't take up, this particular bacteria doesn't take up any plasmid at all. So we basically say this bacteria here, bacteria of this type A, has resistance to neither. So it's resistant to neither antibiotic. So it's resistant to neither of the antibiotics, either streptomycin or tetracycline. Now, so, and most of the bacteria will be like this because the whole process is very low success rate. Remember, it said only 1 in every 1,000 bacteria take up a plasmid. So 999 of the bacteria may be like this, and they're resistant to neither streptomycin or tetracycline. Let's look at this bacteria. Let's say this bacteria is representative of bacteria that take up a plasmid, but they don't take up the recombinant plasmid, they take up this plasmid, take up the non-recombinant plasmid. So let's do this. So bacteria B represent bacteria that take up a non-recombinant plasmid. Okay. Take up a non-recombinant plasmid. I think I'll maybe write this up at the top. It's a bit easier, so I'll put A up. A, B, and C. Let's get a better pen here. A, B, and C. And C. And we'll write this resistant to neither antibiotic, resistant T and T to neither antibiotic. So that's bacteria A, so I'll get rid of that there for a moment. Then I'm saying bacteria B, they take up the non-recombinant plasmid. But a non-recombinant plasmid contains genes for resistant to both antibiotics. So this bacteria ends up with this plasmid then, like that. So it's got, let's see if I can get my genes here really quickly. So it's good. That's the, which one's at the top? That's the streptomycin gene. So it's got the streptomycin gene, and it's also got the tetracycline gene. Yeah. So if we see what it's resistant to both antibiotics. So it's resistant to both. What are these antibiotics called again? TET, T-E-T. I just call that short for tetracycline, and strep. So it's resistant to both antibiotics. Those type of bacteria, which should bacteria B, is that right? And then finally, you have this bacteria over here, and this bacteria, it takes up the recombinant plasmid. So I'll put the recombinant plasmid. So it takes the plasmid, takes up the recombinant plasmid. Now, recombinant plasmid then has a gene for tetracycline, but its gene for streptomycin, put it up the top. I have something to add still. You have to add the fact that it contains the human gene. So it takes up the tetracycline gene, takes up the plasmid, but the human gene is inserted in this case. So it basically takes up the recombinant plasmid. Now, that's really, you can understand how this is quite complicated, and I find this difficult to explain. So essentially, what we're saying is at the end of the whole process, the process being putting human genes into plasmids, and getting plasmids into bacteria, it's all got a very low success rate. Only a few of the plasmids take up the human gene. And then whatever plasmid you have, only a few bacteria will actually take up a plasmid at all. So in essence, at the end of the process, you would have a big beaker full of bacteria. Most of the bacteria would be of this category, bacteria A, and they're resistant to neither antibiotic. They're neither resistant to streptomycin or tetracycline because they've got no plasmid with resistance genes. Then you'd have these group of bacteria. You had a bacteria that did take up a plasmid, but it wasn't a recombinant plasmid. It was one of the original plasmids that hadn't been modified. And these bacteria then might be quite common because there was a lot of plasmids didn't take up the human gene. So you get a lot of bacteria and they would be resistant to both antibiotics because they have a non-modified plasmid. And then here's the bacteria that we're actually after. Bacteria C. These are the bacteria that we've been trying to get all up, and they would be resistant. What would they be resistant to? They're resistant to this gene at the bottom. They're resistant to this antibiotic. Resistant to tetracycline, TET, but not. I got that right. They're resistant, but not streptomycin. TLEP. So essentially, in this third group of bacteria, these are referred to as modified bacteria or genetically modified bacteria, or just modified bacteria. So they actually contain a plasmid, and the plasmid contains a human gene. And we know what do we know about these bacteria? We know that they're resistant to this antibiotic tetracycline, but they're not resistant to this other antibiotic, streptomycin. And that's the means by which we can then locate. We want to locate these bacteria. So in essence, what we do now. So I'm going to take the bottom half of this beaker away now because I need some more space here. So basically, these are the bacteria we want to locate. Bear in mind, they're all in the bacteria. Sorry, they're all in the beaker. I'm going to make us a much smaller beaker here, just to see if I can get the rest of this topic finished, because it's quite, see if we can get the rest of this finished, because it is it's complicated and difficult. So give me just a second if I get my duster. So in essence, there we are. What we want to do now is we've got this, make it a much smaller beaker now. We want to basically locate these bacteria. How do we locate these bacteria? Well, you'll see then that there's a good illustration of this in question 4. Question 4 uses a different antibiotic resistance genes, but essentially it's the same. It's the same plan. And you'll see what you do now is you then start making agar plates. So you start off with an agar plate, or a petri dish if you want to call it, and it contains agar. So there's the agar. And what you do is you just prepare an agar plate like that, and that agar plate you put into it agar, and that's all. You just put in normal agar in which bacteria would grow, and in this plate you put no antibiotics. Now, but no antibiotics at all. You don't put either tetracycline or streptomycin in the agar, so there's no antibiotics in this dish at all. The question is, and then what you do is you take a drop of the liquid from the beaker, and the beaker, bear in mind, contains a mixture of these bacteria, and among the bacteria there's this kind of bacteria, the one that you want to modify. So you take a drop of this liquid and you transfer it onto the surface of this petri dish, where you allow the bacteria to grow. Bear in mind, this agar contains neither antibiotic, no antibiotic at all, and you put some of the liquid from the beaker onto this petri dish. Question is, which of these 3 types of bacteria would grow in this petri dish? And the answer is all 3 of them. You might say, well, how would these bacteria grow in this petri dish? Well, they'll just grow. They grow in the petri dish. So all 3, A, B, and C, all 3 types of bacteria would grow in that dish. Because there's no need for any antibiotic resistance genes to grow in this agar, because there's no antibiotic present. That makes sense. So all 3 bacteria can grow in that dish. Now, what you then do is you make up a second dish. And into the second dish you put agar again, is the agar. But this time you add some antibiotic. And into the second dish you would add the antibiotic streptomycin. Sorry, tetracycline. So you add in tetracycline, T-E-T-R-C-Y-C-L-I-N-E. Tetracycline. So you add some of that antibiotic into the agar. Now, the question is, which of the bacteria could grow in this dish? Because what you do is you transfer the bacteria from the first dish to the second dish, and you do it by pressing a piece of paper down onto the surface of this dish, and that picks up some of each of the 3 types of bacteria, and then you transfer them to the second dish. You press the piece of paper down onto the second dish, and that transfers the bacteria. Question is, which of the 3 types of bacteria would grow in a dish that contains tetracycline? Well, the answer is not A. Why will A not grow in this dish? Well, A have got no resistance to the other antibiotic because they've got no plasmid. So A won't grow. What about B? Well, B's got resistance to both. Resistant to both, so it's got resistance to tetracycline. So B will grow here on this dish. And C, well, C's got resistance to tetracycline. So B and C will both grow on this dish. Okay. And then here's what we do, prepare a third petri dish. Agar in it as well. And into this dish we put streptomycin antibiotic. And once again we transfer bacteria from this dish to this dish. We transfer B and C, and the question is, which of B or C would grow in a dish that contains streptomycin? Well, B will definitely grow here, because B's got both genes, so it'll grow in this third dish, because it's got resistance to both these antibiotics. Let's just put that back again, accidentally touched that. So bacteria B will definitely grow on grow on this dish. What about C? Well, bacteria C, these are our transformed bacteria. They can grow on the tetracycline dish because they're resistant to but they're not resistant to streptomycin. So they can't grow on this third dish. Now, I hope you understood all that, or you were able to follow that. It's really, really difficult. And here's the thing, and usually I say this to students at the start of this explanation. I say to them, look how many marks are for this. And if you look at question 1, and you look at the relevant part of this question, it's 1C part 2, and you see that there's 2 marks for this. 2 marks for an explanation of all of this goings on. 2 marks for it. And if you look on the essay question, you'll probably see that there's possibly only 2 marks allocated to this as well. That's the essay question 9. And also to another question, I think it's question 4. Essentially just 2 marks. So really all that explanation for 2 marks. So here's my advice. Don't be getting too worried about this. Don't be trying to learn this diagram or understand this diagram too well. I would advise you, if you get some basic understanding of what's going on here, then that's okay. But be able to come up with 2 marks to answer that question. And I'm just going to tell you what all this is. Well, how I deal with it in the notes there is, if you look on page 8 and page 9, the bottom of page 8, I say, at the end of the whole process, scientists will end up with 3 types of bacteria. Bacteria with no plasmid, and they're susceptible to both antibiotics. Bacteria without the non-recombinant plasmid, and they're resistant to both antibiotics because they've got the 2 genes for both. And then the third bacteria, these are the transformed bacteria with the recombinant plasmid. These bacteria are resistant to tetracycline but not streptomycin. Scientists want to be able to locate these bacteria. How do they go about it? Here's the process. This process, for 2 marks, you start off and you say, scientists do a process called replica plating. This process is called replica plating. And basically you're just referring to what's going on with the agar plates here. Replica plating, it's called. And if you say that, basically what you say, set up a series of plates, one without any antibiotics present, one with tetracycline present, and one with streptomycin present. Transfer bacteria onto each of the plates. That'll get you the first mark. So basically, if you mention it, you say this is called replica plating. Transfer bacteria onto each of the plates. And then the second mark is locate those bacteria that can grow on the tetracycline plate, but can't grow on the streptomycin plate. So you're looking for those bacteria that are able to grow on the tetracycline, on the agar containing tetracycline, but they can't grow on the plate that contains streptomycin. In other words, we're looking for, those are the bacteria. So bacteria that can grow on the tetracycline resistant, on the agar with tetracycline, but can't grow on the plate with antibiotic streptomycin, they are the bacteria you're after. So once you locate those bacteria on this plate, you can then scrape those bacteria off the plate, and you now know you have a sample of modified or transformed or genetically modified bacteria. And you'll see there, having gone through all of that, at the bottom of page 10, I suggest that you just know those 2 points. At the bottom of page 10, it said the exam question, create 2 replica plates to which streptomycin or tetracycline has been added. Colonies, for the word colonies, you'd use bacteria that grow on the tetracycline, grow on this one, but don't grow on the streptomycin, are the transformed or modified or genetically modified bacteria. And that gets you your 2 marks. That gets you your 2 marks for that particular question. That's question 1C part 2. If you're looking at a similar question, this is a much older question, question 4, and you see essentially it's the same. It follows the same stages as question 1. It asks part 1. It asks A, how is the section of DNA been cut out of the human chromosome? You would say it's cut out with the restriction endonuclease enzymes that have sticky ends, cut with sticky ends, and then it shows. It uses different plasmids. It uses a plasmid containing an ampicillin resistance gene. Ampicillin is another antibiotic, and a tetracycline resistance gene. And you'll see in this case that the human gene is inserted into the middle of the tetracycline resistance gene. And so if the bacteria that are successful will contain this modified plasmid, so they should be resistant to ampicillin, but not resistant to tetracycline. Essentially, that's the same thing. Part B of that question says, how would the section of DNA been inserted into the plasmid? In other words, how do you get the human gene into the plasmid? Answer is, cut the plasmid open with the same restriction endonuclease enzyme that you used to cut out the human gene, so that it has matching or complementary sticky ends, and then insert the human gene into the plasmid using the enzyme DNA ligase. Over the page, this question follows question 1 in the same format. Describe how bacteria may be induced to take up the plasmid. Think of the previous question said, how would they be encouraged, induced? And the answer is, use ice cold calcium ion solution and heat shock. That gets 1 mark. And then it gives quite a nice illustration there. This question is quite good to follow this process. You'll see it shows the 3 plates. It shows a plate, nutrient medium only. I always get students to write on the nutrient medium only. I get them to write no antibiotic, neither antibiotic. And you'll see this plate then, you see lots of bacteria growing on this plate. And this will represent this plate here. And basically all those bacteria, they're all numbered in their positions. And these are all, so it's from 1 to 12. So they'll contain all the different groups of bacteria because there's no antibiotic. You'll see that on the second plate, you've got bacteria that can survive ampicillin, and so that would be B. In this case, similar train. And then you see when you put them onto a plate with tetracycline, you can see that bacteria 4 and 11 have survived ampicillin but not tetracycline. So bacteria 4 and 11 must be the transformed bacteria. That's D part 1. Why the agar plate containing nutrient medium only? In other words, no antibiotics, contains the greatest number of bacterial colonies. I would just say it doesn't contain any antibiotic, and therefore all of the bacteria, whether they're transformed or not, can survive on that plate. And then it says, which colonies contain the bacteria which have taken up the transformed plasmids? And those are just 2 marks. And the answer is colonies 4 and 11, because 4 and 11 must contain an ampicillin resistance gene, but they don't contain the tetracycline resistance gene. Why do they not contain the tetracycline resistance gene? Because that's where the human gene has been inserted. So these are the modified bacteria. So the modified bacteria will contain modified plasmid, so they'll be resistant to ampicillin but not tetracycline. So you see it follows the same sorts of stages, as question 1. Only other thing, question 1, and I'm going to finish here just now. 1C part 3. It asks a question, and after that it says, how does the elimination of the other bacteria, in other words, how does getting rid of these bacteria and just singling these ones out, how would that increase the efficiency of production of hormone? Well, we should then talk about the hormones insulin. Basically what we do is, once we have established that bacteria C is the transformed bacteria, we take these bacteria and then we place them in a big tank. It used to be called a fermenter tank, and you give them lots of glucose and nutrients and everything, bacteria C, and you allow them to multiply up them. And all of these bacteria C contain the insulin gene. And so you get multiple copies of millions and millions of bacteria that contain the insulin gene. So you basically want to isolate these ones out and put them into the fermenter tank. You don't want any of these others. You don't want B or A. And that's basically what that part 3, it says, how getting rid of A and B, how getting rid of bacteria that are not transformed, How does that increase the efficiency of production of hormone? Well, you could argue that if you let all the bacteria into the fermenter tank, then they would compete, all these other bacteria that aren't making insulin, they would compete with the modified bacteria, and so you get less insulin made. So basically that's it. You would say you're reducing, by eliminating all the bacteria, reducing competition for the resources in the fermenter tank, during the production of insulin. So I'm going to stop there. By the way, this is really difficult. Students find this very difficult, even after I've explained it. Most students still say, right, that's just a bit too difficult. But what I would suggest is, no, there's just 2 marks for it. So just learn the 2 marks. So the 2 marks are, again, this is called replica plating. So you set up, I would just say it this way, set up agar plates that contain no antibiotic, tetracycline antibiotic, and streptomycin antibiotic. Set up those 3 plates, and then grow the bacteria on those plates, and isolate those bacteria that can grow on the tetracycline plate but can't grow on the streptomycin plate. In other words, you're isolating bacteria C. B must be the transformed bacteria, and essentially that's it. I've never seen this explanation required. You never have to go into it or explain. And even there in question 4, where they set out a sort of illustration of agar plates and so on, they don't ask you very much detail. Essentially just ask you how does this process work. So there you are. That's as far as we're going to get in this section. And really all we've done is we've kind of just looked at transferring genes into bacterial cells. We've made, you refer to the bacteria then as transformed bacteria, or genetically modified bacteria, or genetically engineered bacteria. There's a lot of words in this topic. In the next video, I'll try to move on to look at genetically modified plants. In other words, putting genes into plant cells, and genetically modified animals, and then also look at gene therapy, which is inserting genes into humans to correct some disease which they have due to a genetic abnormality. So, for example, cystic fibrosis, where you put the proper gene or the functional gene into an individual who has cystic fibrosis. So there you are. Thank you very much, and watch out for the last video in this section. Okay, thanks.